All right, so again, welcome again. Today, as we mentioned, part 2 of the ST basics. So really just trying to do a few more examples, a few more topics that we didn't get to last week, showing some more treatments and assessment examples as we build our criteria, and then talking about some of the reporting analysis options and the graphing functionality that's in ST. So that is our target today. Just a real quick recap of the most basics that we covered last week. And really in ST, we're dealing with criteria files. That's really what we're creating is a criteria, and that really is just a question. Is really what we're formulating, or maybe a more technical term would be a query. Would be both terms that really fit what it is we're doing in ST. And that question has 3 parts that we talked about and showed, where we select certain trials that we want to be searching across. Then we specify which treatments that we want to include in our summary. We started with the full list of all the unique treatments that were found in all of our trials. And then we could whittle it down or be more specific and do some things with that list to kind of focus in on what we care about. And then finally, the third part was what to do with the columns of data for analysis. Which ones we care about, which we don't, how should they be combined? We had talked about, it's almost like a deck of playing cards that you can sort into 2 different aspects, whether you put all the even numbers in one group and put all the odd numbers on the floor, or things like that. And that's really what we were doing with our criteria in the assessment section, was giving instructions on how to put together, which columns to leave out, things like that. And then we just looked real briefly at kind of the analysis, the output that we were getting, and how that looked, and then the report that was generated. So today we're going to try to touch on really each of those subjects again, just in a little more detail. And I have more or less the same set of trials that we worked on last time, but rigged up to be a little bit trickier. Last time things went very smoothly, especially in the treatments editor. And so I have kind of planted some fake issues, if you will, to work through. So let's dive in. Here I am in ST, and we'll just do like we did last time, starting with a new criteria. And we choose that standard definition that we do, just like we do with our protocols, the GAL7, and I'm going to select my trials to search from my study list. Last time we used the tutorial list, and we use these ST exam files. Here I've resaved some of them, like I said, to kind of plant some issues. So you may not be able to follow along directly with these, since these are a little bit messed up. Compared to last week, where we were using the exact same files that everyone has on their computer. Hopefully we'll be able to illustrate kind of what the differences are. So we've got those 6 trials that we care about. And I'll just proceed through the settings. And then we're taken to the treatments editor, and we'll do that build treatments functionality again to define, to give us the list of all the unique treatments that are found in these 6 trials. As I mentioned last time, things were really smooth. It really had just a very short list because all of the trials came from the same protocol. And so they all had really identical treatment lists. It was very easy for us to work with. But here now we're going to see that maybe it's not always that easy. Still choose treatment name and our rate and rate unit. We care about those 2 both in order to get our unique list. Okay, So here I've got 8 treatments this time. If you recall last time, I think we only had 4. And you can actually see we had 4 come from exam 2 and then a whole nother set from exam 6. So I think this last trial was the one that I made kind of messy to illustrate some of these points here for us. So really, I think that first step after building those treatments is to just kind of review and look to see what it is we have and what is, what's the difference compared to what we want. What are we trying to focus on? And so treatments 1 through 4 look pretty much like last time. We have these make-believe herbicides or fungicides, I don't remember, but these fictional products that were being applied on our plots, and then we had an untreated as well. I think this is an adjuvant or something, some additional product, and then an untreated. But now we have some other examples that didn't quite combine as nicely. And in this case, I see it's called Super Stomp, just like treatment number 1, and it's even applied at the same rate. So to me, it looks like it's the very same product, but the only difference is it looks like there was a slight difference in how it was entered in the treatment name. So this is a perfect case where someone must have hand typed, they didn't use our list or something like that. And so although it really truly is the same product, the name was different and therefore it counts differently. And one of the downsides, I guess, to computers in general, they do exactly what you tell them to do. And in this case, we told it that the treatment name is, if it's different, it should be a different product. So the way to work around this, and we did an example last time of this, is if we go all the way to the right in the treatment editor, there's these extra columns that we typically don't see in ARM, but they are helpful to us in ST. And we're wanting to use the equivalent treatment. So if we look at this treatment number 5, we would say that really it's the exact same as treatment number 1. So if I encounter this super dash stomp anywhere when I'm wanting to run my summary, I really want it to count the exact same as treatment 1. I want it to be combined with treatment 1. So in the equivalent treatment column for this row, I'm going to put a treatment number 1 here. So now this will combine them automatically. So that way we really don't have any data for Super Stomp with the dash, but instead it'll all be combined together with the treatment 1. So that's going to allow us to have that control and deal with the fact that sometimes things aren't quite entered the same, even though they really are the same product. Looking at this next treatment, I see Stamper 123 is the name of this product, and we have that adjuvant, whatever that other product might be. Really the difference, I'm comparing that to treatment 2, actually looks very similar. The main difference being Stamper versus Stamper 123. So of course we could continue to use the equivalent treatment if we wanted, and just kind of hard code everything in like that. But say maybe there's a few different versions of this stamper. For whatever reason, they had different names, but really maybe for our purpose, we want them to be combined together. They really maybe are the exact same product, just they were entered differently, or what have you. So another tool in our toolbox for the treatment editor would be to use a wildcard. We had done a couple of those in the assessment section last time around, but we can also use some of them here in the treatments editor. And what we can do is, considering the stamper and Stamper 123, if I put an asterisk at the end of this stamper, what that's going to do is match, and you can actually see on the ST properties that it changed the selection over here to say that as long as the treatment name starts with Stamper, then anything else after it, so that star asterisk wildcard is anything else essentially, then they'll all be combined together. So whether it was just Stamper or Stamper 123 or Stamper 762 or whatever the other name is, as long as the name of it started with Stamper, then it's going to match with this treatment number 2. It'll still have, since we have this adjuvant here, it'll also include that criteria that it would have to start with Stamper and then also have the C50220 product with it. But as we compare that treatment 6, we can see that that really will be the same thing. So using this asterisk, or you could also go on the ST properties, and if you change this, you'll see whole field was what we were originally, but if you change the selection to start a field, it'll also give you that same thing. So if you're not super familiar with those wildcards and feel a little more comfortable just kind of reading this, it does the same thing. I want to match based on the start of the field, or I want anything that's not Stamper. There's a different wildcard for that. Within puts asterisks on both sides. So if my product was 321 Stamper, it was the name of it, but I really wanted it, those are really the same, I could do this within and have an asterisk on both sides. So a few different ways to leverage those wildcards, whether you kind of know that syntax and you're comfortable with it, you can type it in, or here on the ST properties panel to the right, maybe a little more intuitive way to interact with that matching. So that basically replaces this treatment number 6 then. If we define this correctly, then the Stamper 123 will be included with our criteria for treatment number 2. So now we may as well just delete this treatment out of here. And again, we did this last week, and the important thing to note is right now we're only changing this criteria. We're not changing anything in our 6 trials. We're not deleting treatments. We're not removing data. We're just changing the question. So we'll say we don't care about that one anymore because we know we've included that data with a more powerful criteria, I guess you could consider it here with treatment 2. One tip here I have in the questions here, there's a lot of columns on the treatment editor, and I've been scrolling back and forth. We can actually use, just like in ARM, we can use views to hide some of those columns. And at this point, I think that would be very helpful to see that other information. So here in my regular properties panel at the bottom, just like we have in ARM, we can turn on or off certain fields. And I want to hide empty fields. And that should take most of them away. And it actually took out everything I didn't want, but kind of nice, it does leave those 3 special columns visible still for us, which is nice because since in ST, they're kind of special and we want to make sure they're still visible. So thanks for that tip. It should hopefully make it a little easier to follow along instead of scrolling so much. Next example, like it's still treatment 6 since we deleted the last one. We have a sure kill, just like our treatment number 3, but this time there's a slightly different adjuvant that's going with it, or whatever type of product this is. So sometimes you might really have just a focus on this, for example, our sure kill product, and we really don't care what other products might have been with it. Depending on where the trial was, you might just have certain standards or whatever product was available to go with the trial. Then we really don't care. We really don't want to have to deal with all those other different types, all those different combinations, when all that really matters to us is, product sure kill at 2 liters per hectare, and that's all that matters. Everything else I want combined. I want to combine any scenario that has that sure kill, and it doesn't matter what else is with it. So in a situation like that, there's a wild card here. I think what I'll do is just insert, maybe I'll clear this line out. I think that'll be the most clear thing to do. In order to do that, kind of with any, I guess would be kind of what I call this wild card. Just create a new line to go with that product that you care about. And then we're just going to use 2 asterisks, specifically in the treatment name column. So now for criteria treatment number 3, I've got my sure kill product that I care about, and then a line that simply has those 2 star asterisks. And that's going to combine anything that has sure kill at 2 liters per hectare, and then whatever other components might be found in any of my trials, doesn't matter. They're all going to be combined together. So the example here where we had this C60,000 versus the previous one that has sure kill with the C50,220, both of those are going to be combined together. They're both going to be matched in this criteria here, because it doesn't matter what other products were with sure kill. This is the one we care about, and we want to bring in everything that has that product, at this rate. So that's the with any wild card. Again, the important thing is to put it in the treatment name and have that same treatment number with the product that you care about. All right, so that really takes care of treatment 6. So I'm going to delete that treatment out once again. So you can see we're kind of narrowing down our list of treatments. And then the last one, a pretty straightforward one, we have a check, whereas in our other trials, we were calling them untreated. That's a fairly common one. And of course, we can just do an equivalent treatment on that. Oops, I don't want to say number 1, it's equivalent to number 4. And we can mark those as the same. Now, something like that, though, of course, is maybe so common, and it happens all the time that you're doing this. We do have a synonym list. Don't really have time to cover all of the functionality with the synonym list, but just kind of want to point out that it is there. So if I right-click in the treatment editor, doesn't really matter where, you'll see towards the bottom, there's some references to the synonym list. And I'm just going to click Edit Synonym List so you can actually see what this is. Essentially, you're creating a group of treatment names that are all really the same. So in my case here, I've got UTC, untreated, CHK, check. Maybe those are all the different types of ways that I typically see the untreated check be put in trials. And so putting these all in a group or this synonym list, then ST can automatically recognize for us those similar items and just automatically mark them as equivalent. So instead of us manually having to see this and go in and mark it as an equivalent treatment, the fact that I have both untreated and check in my list in the same group ST will be able to recognize and mark those as equivalent. So all I have to do in that scenario, I just need to right click here, and there is a command to set equivalent treatments from our synonym list grouping. And there it's going to pop up my synonym list. I could turn off certain ones if I was concerned about it, but here it already had recognized that check is possible to use here. So I'm going to say, yes, that's definitely the group I care about. And the screen needs to refresh, or what I did there. Let me try that once more. Let me just turn all of these on. I'm not sure why it didn't. Okay, well, it's not marking that. Let me clear this. Not sure what I'm missing here in my steps. Set equivalent treatments. It's supposed to fill in our equivalent treatments for us, and I'm not sure if I'm just missing a step or what I've done here. All right, well, that's what I get for trying to show that. I'll play around with that and see. I must have just missed a step, but in theory, if I did it correctly, it would recognize these two are the same, and it would put in this case, it should be a 4 to mark this check as the same as this untreated here, to treat them the same. I'll have to brush up on my equivalent treatments to see what I missed there, but at any rate, that is kind of a little bit more advanced function in ST, but definitely one that can be useful. The main reason, the main difference between the synonym list and just using equivalent treatment all the time is that synonym list exists across all of your STs, across all of your use of ST, whereas, marking these as equivalent on the screen here is only for this one criteria. So the next time I'm wanting to summarize a different set of trials, I have to do that manual process of marking things as equivalent again. But that synonym list really exists now across all my criterias, and I can just set equivalent treatments from that. So it's possible to add your own set of products as well if they're ones you work with often, assuming you can get it to work better than I did today. But anyway. That kind of rounds out our treatment editor here. Again, just showing more examples of when things aren't quite perfectly cut and dried in our trials, that we have the ability in ST to overcome some of those inconsistencies. And it might be kind of a balancing act. You of course could always open up your individual trials and clean them up a little bit as well. So sometimes it's kind of a balancing act of what makes more sense. Should I, open up all 50 of these trials and make their treatment lists, really, really consistent? Or, do I do some of these tricks here in the criteria file in order to get things to match the way I want? Kind of just depends on your situation and what makes the most sense. But you have options either way. Have a tip on my synonym list. Maybe it's this. Yeah, it looks like I probably hand entered these playing around with another scenario. I had these ID numbers in here, and maybe that's probably exactly what, because they probably shouldn't be filled in there. Now let's clear that out to see if it'll actually do that. Nope. Darn. All right. Anyway, I'll have to figure that out later. See what I did. But thanks for the help on that. All right. Moving on to the next section. So we kind of covered treatments. That final the final section we at least totally need for our criteria is the assessment section. And so last time we had a scenario, and I'm going to kind of kickstart with that. We were really looking at our control ratings. And I think we specified a rating unit, although I'm not sure if it mattered in the end. We chose multiple rating units. So this was really focusing in on only assessment columns with a control as the rating type, and then a rating unit either in percent or %UNCK, with the assumption, that we trust that those two really are the same type of assessment. Sometimes you have to be careful with that if you're having calculated columns, things like that. So we had really done looked at that, and originally our first scenario we only included that in our criteria, and we had just tons of data points all combined into one, and that seemed surprising since we only had 6 trials. And so we came back and we used the match and separate wildcard, this little @ symbol, and that split everything up by the different pests that were found in the different trials, which in our case really was important, because otherwise we were combining, all these different analysis or results from these different pests all into one big group that really tells us nothing. So instead, we were able to see the different split outs from that. So that's really where we started from. We didn't do anything with the interval, and so we had a lot of the things split out. So kind of building on that idea, we had a lot of different grand mean columns. And actually, let's just real quick, let's do the begin summary on that and make sure we're in a good spot with at least approximately what we were getting from before. So we've got all those assumptions of AOV. I just ignored those error messages because now that we're combining all this data, a lot of that goes out the door anyway. But we matched everything, so we didn't mess up our criteria in that regards. And so here I've got, I have so many header rows here, but we have that same control rating just with a bunch of different pests. So some of them, like common lamb's quarter, we had some from all of our trials, some not so much. So crabgrass was good, some of them maybe didn't. So that's a nice starting point really for us to see all of that matching. But one thing we could do is maybe focus in a little. So that's all great, seeing all of those different pests that we were looking at. Alternatively, we could, focus in on one particular pest. If that was, maybe we want to create a summary related just to that. We don't need all of those other ones. We can certainly change that criteria. Maybe I'll grab the barnyard grass and choose that specifically. And it doesn't take long. I'll just run this summary again just to show that really we're going to toss out all of those other columns. So now we only have one set really of data. We've got this grand mean with the barnyard grass. And then to the right are the individual columns from each trial. It looks like at least 3 of them had this common barnyard grass in it. Somehow I have a second grand mean column. I'm not sure why this one is different. So I usually like to compare the kind of the header description to see what was different. It looks like my crop type, for whatever reason, this has a C in it and that one doesn't. So kind of the typical workflow with with ST is kind of on display here, kind of going back and forth of why are these not all being combined together? And I realized really the only difference in this description versus this description is this crop type. So some of these trials, the crop type was empty, and some of them had the C filled in. I'm guessing it's because these trials are really old, and I just haven't actually been in them long enough to have that automatically refresh. But if I close that, then it's just a matter of going to crop type, and I can change that from match and separating just to the asterisk, or even clear it out if I wanted to. And if I just keep running that summary, we can see if that small change gives us the results we want. So you will find a lot of back and forth. All right, but that looks good. Now we are down to just one grand mean column from all of those assessments combined together. So the other big example I wanted to show, and as we look at what we have here, I notice we have multiple columns from a single trial. And if I compare, I can see it's because we went out and performed this assessment at more than one date. So that can really be important for us, is separating the time of year. At what point during the season was the assessment done? Is another important aspect, potentially, if that's something you would care about in your criteria or in your summary. So there's a couple ways to do this, but one way that definitely doesn't work would be using the rating date. Naturally, every single trial is going to have a slightly different date as to when the rating was done. That's not going to be very practical for us to work with in our criteria. In theory, it technically would be possible, I guess, if we hand chose each one. But what makes a lot more sense would be to utilize some of these interval fields that ARM automatically calculates in your trials. So in an assessment, as soon as a rating date is filled in, ARM calculates these days after first and last application, the treatment eval interval, days after emergence, things like that. It just automatically calculates relative to those other dates in that trial. And so that can be really powerful for us to use as a way to determine when the assessments were done. In our case, it kind of looked like we had an early and a late, essentially, assessment, one that was pretty recent relative to when we performed our application, and then we came back, several weeks later to see the results again. So maybe we want to split those out and look to see the analysis on the earlies and then the analysis on the lates kind of separately. So in my case, I'll use the treatment eval interval. And you'll notice when I click into this field, I don't have a gold box. When we were in like rating type, we did, but the interval we don't. And if you don't have a gold box here on that particular field, we can use the ST properties to kind of build that gold box, if you will, of being able to choose particular ones. And we'll see why it's different in a moment. So using this possible criteria section over on the properties panel, make sure first that you're clicked into that interval field. Then under possible criteria, there's a button to scan for criteria. So ST will open up all those trials and give us a list of all the unique treatment eval intervals in all the assessment columns. There's a box here to match existing entries. So this will actually only give us the unique intervals for the columns that we were already matching. So in our case, only for those control ratings with the barnyard grass. So only the ones that were already included in our analysis based on what we have already defined in this column will be actually checked here to give us our unique list. So that might be helpful for us, I think, especially in this case, to clear out some of the noise, if you will, of those other ones. So you can see real quick, it opened each one of those trials just long enough to fill in that list. And so that list is actually here. Let me minimize that again. Again, on that possible criteria, now we have a drop-down list. And we can see now the list of unique intervals. And just like we saw a moment ago when we were looking at our criteria, we can kind of see a clustering. We've got 3 different dates that are all clustered around that 2 weeks after our application. And then we have a set, a couple weeks after that. So what I can do is select these 3 and actually make them into a range or make them into a group. So if I click on the range button, that will then set 13 through 17 days after application B. So any assessments that fit within that range will be matched together for us in the assessment. Now, ST wants to know if we want to select more ranges. And I want to say yes, because we had that early set. Now I want to do basically the same thing, but with that later set of assessments. So I'll select these 2 and click range. And now I can say, no, those were the only ranges I want. So now what happened is that first criteria that we were working on, we have that range of 13 to 17. We'll combine all of those assessments together. Then you can see ST copied our criteria. It's going to do the exact same type of query, only difference being the range of intervals. And now we have that later set of assessments. So if we go to the begin summary, we can kind of see this in action. Now we have our control ratings, but we have only 1 from each of those 4 trials that we're matching because we made that specific group of early assessments. And so we've really focused in on our, what we want to see. So we can get a good idea of how our products fared against each other in the early assessment. And then if we scroll to the right, looks like this very same number of trials matched with those late assessments. We can see there actually was slightly different performance between the 2. But again, the main idea is really when working with ST to not just see this analysis and say, great, it gave me something, here's my results, and let's go. It's important to take a look at that count, take a look at the trials that are being matched, and think, is this what I want to be combined? Is this make sense? What is being put together for these means? Instead of just blindly accepting whatever we had. Because when we first started this, it was a mess. It was everything combined into one set of data. And now we've grown and built that criteria to be a lot more nuanced and tell us exactly what we want, which in this case is, how did our products do in controlling our barnyard grass, our weed of interest, in these trials, both from an early and a late perspective. Before we move on, just one other quick note kind of on that same idea of the timing. We use the treatment eval interval. You certainly could use some of these other calculated columns, just depending on your trial. One other option that would be possible is utilizing the rating timing field. So this is just the regular list, as if you were in a trial and choosing a rating timing. It's just a very arbitrary schedule, essentially, of the assessments in a trial. So, whether you already use them as kind of part of the scheduled tasks feature in ARM trial, kind of hooks up to this list, or kind of after the fact, it can be a tool for ST as well to go back into the trials and put in, an A1 to match up with your early assessment. And then your A2 could be the late assessment. So instead of, if you don't really have a treatment eval interval, or some problem that makes it hard to really query on one particular field, you could always kind of hard code, if you will, your rating timings to be, these are the ones I want, and then in your criteria do something like that. And you can hard code then that A1 to grab any columns that had A1 filled in. So just another option. In our case, that wouldn't work because rating timing was empty in all of our trials, but that's kind of one more opportunity, just depending on what you have in your trials. Okay, so that covers the assessment section. Next thing I want to cover would be the reporting options. And you can get there either on this begin summary, there's the summary options, or if you already had gone farther into the analysis, we can also get there here while we're reviewing what we've received. There is the options button here. Either one will take us to this screen here. And we covered these really briefly before. There's really three main sections here for ST. And just starting maybe with this first ST summary options, there's different options about your treatment matching. You can group your means if you wanted to. I think the most common options we'd be looking at, your mean comparison test is here. So we have similar options to what we can run in ARM, also in ST. These first four here are, in order, just like we have in ARM, of how liberal or conservative they are. And then you can also set your significance level as well. There are a couple of options here for controlling whether those mean separation test is even performed. If you were with us on some of the statistics sessions we did earlier, we kind of talked about this option, maybe looks familiar from ARM side, of not running the mean separation on the individual trials if only when the AOV analysis is significant. There's kind of the same idea for the grand means, only if the treatment by trial interaction is not significant. So it's kind of the opposite in this case, where if there is a significant concern for interaction, then maybe it's not safe for us to draw conclusions. And that's really the idea behind that option, is if that treatment by trial interaction is a concern, then we maybe don't want to just start drawing conclusions and giving those mean comparisons, but instead we should maybe look into that a little more instead. So that option is there. And we saw that last time when we went into the report. We have the AOV table that splits out all the different sources of variation, and one of them towards the bottom was the treatment by trial. And if that p-value is significant, then there's a concern. And we'll see a graph here real soon that we can use to kind of dig into that further. The other option I want to mention here related to the analysis is this AOV method. There's not super, I guess we've got advanced, combined, and classic, but they're really related to the ST versions, which mostly just says how modern it is, I would say. So you really want to run the advanced, being the most, the most recent, most modern way to approach things. These other two would really only be if for some reason you were trying to exactly match some report made in 1998 or something like that. But the fixed effect here is fairly important to consider, what to choose here, because it can have an effect on your analysis. And really it comes down to your criteria and your trials that you're summarizing. Usually treatment is the one you'd want to select. And the reason being, a researcher has more control over the treatments, like, getting to choose the specific formulation concentration to follow, than you do controlling the trial conditions, the soil, the weather, all of that stuff. A lot harder to control that compared to the control we had in choosing what treatments we were going to include. So, really, if your goal for your criteria for your research is to generalize across the trials, like, is this super stomp product effective in most fields, then that would be why you would choose treatment. So that's usually the option there. If you go into the help, it'll maybe talk about why you might choose trial or might choose both. But yeah, in most cases, treatment usually is the one you would choose. Real quick, looking at the means here, mostly this is just turning on or off some of that information that we saw before. So like minimum and maximum standard deviation, maybe I'll turn a couple of them off just to see how it'll shrink down the report and give us just fewer numbers, fewer fluff, maybe a little easier to read. So you can turn on and off different things. For the grand mean columns, those were those blue ones that summarized everything, and then the individual trial columns. You can turn those off entirely if you wanted. Then the format tab is just kind of what is being included, which header rows do we want to be included. That's the description. We have a little number next to which line number that is in the header. You can choose which ones show up, things like that. And then there's the graphs, which ones should we include in the report itself. So if I change some options, that really would affect the analysis. It'll rerun the summary based on those changes. And now we can see our difference. Here really it's just we took away some of those numbers, and it is just a lot easier to read, I think. Finally, I want to make sure to cover the graphing here. The graphs automatically are included with the report when we press next, but we can kind of interact with them a bit by clicking this graph button. This will create labels for us, so I'll just take the default. The header fields for the left title, let's just do control, maybe just the reading type. And I'll just leave the right title blank. Just had a webinar on the graphing tool, actually. So you can check out the recording there for all of the options. This really is the same functionality, the same options that we have in ARM, we have here as well in ST. So hopefully it's familiar to you. But what we have, a few different graph types in this case. This first one, we are looking based on treatments. So treatment 1, 2, 3, 4 in our criteria, and then we can see the results. My axis here is not very helpful. Let me go back to that zero origin there. Now it's a little more interesting. There's not much variance here, so we don't see a whole lot, but this is based on each treatment. Then we're combining across the trials. You can kind of see the other cross section of trials. So this would be, you can see on the right which trial ID that is, and then we're combining across all the different treatments to see the results here. And then we have the treatment by trial interaction. And I think just because I have kind of an unusual resolution here on this screen for presenting, it's showing up really small. But this would be the tool to use when we had that treatment by trial interaction issue. I might see if I can bring up the help real quickly, because I think I have a picture in the help treatment by trial graph. There we go. This will be a little easier to view anyway, and a nice example for us. Just real quick, kind of how to read this. There's a lot of things going on with this graph, but the top half is the treatment stability. And so we're graphing treatment means within each trial against the overall trial mean. So really what you want to look at is the slope of each of these lines. So each line kind of corresponds with a treatment, and we're kind of comparing the slopes of those lines relative to each other. So our x-axis is the different trials, like the overall mean for each trial. So trial 1 is here. Trial 2 was pretty high. In this case, this is yield data, so pretty high yielding, whereas treatments 3 and 4 were low yielding. So we're kind of comparing the slopes. If you have a high slope, so the green and the blue here, that would indicate that those treatments are more sensitive to the trial effects. In this case, yield. So a high, in a high yielding trial, these products did very well, whereas in a low yielding trial, these did poorly. Compared to this red slope that isn't quite as steep, it was more consistent then. So yeah, it didn't do fantastic in the low yielding, it didn't do super in the high yielding, but it was very consistent then throughout. So you're kind of comparing those slopes, again, depending on what data you're looking at. In our example that we have been working with, that was actually control. So it might be kind of the opposite, that you're hoping for a better performance, you'd be having a low number. But here, this is yield, so higher the better, in the performance at least. So that's just kind of looking. Obviously, this bottom one was the check, so it was always 100, and then these were percent of check. It looks like percent relative to the check. This bottom half is a dendrogram, really clustering the trials based on their performance. So you can see 3 and 4 were right next to each other, so they have a close grouping. Treatment 1 is also close enough together that they all kind of represent this half here of the low yielding trials. And then these 3 over here really end up being the high yielding, because they're all in this second bracket. And so really what overall you're comparing the slopes up above here, and if they are really different, then you're concerned about that interaction. If most of these slopes are pretty much the same, then we probably don't have any problem with interaction. So we could use our report, that p-value of the treatment by trial interaction, as kind of a nice decision maker there. Is that significant or not, to really decide for sure, do we have a problem or not? And if there is interaction, then maybe we're better off splitting up and using this clustering to maybe only analyze the high yielding. So maybe let's select only these 3 trials and do a summary, and we can then kind of make some conclusions there, and then take these other trials, the low yielding, and draw some conclusions there, because maybe they're a little different. So that's a quick rundown. There's a whole lot there. We could spend an hour just talking about that idea there, but hopefully the help here kind of splits it up. If you do want to learn more and go into more detail, this help topic tries to talk through each step, kind of like we did here, maybe just a little slower and more helpful, and uses this example to kind of talk through it. So that's a real quick look at diving deeper, I guess we could say, into the analysis. All right, right on time. We just hit the hour. I can stick around for questions. I try to leave a couple minutes, and I always get long-winded, so apologize for that. But thanks everyone for joining us today. I will admit ST is really hard to train on in the fact of trying to create examples that make sense. So I typically tell people to get in with your own data and play around with it a little bit. It becomes a lot more real, a lot more alive, and that's probably when questions will come up, and definitely use us as a resource. We send an email to our support box, and we can help set you in the right direction. But it's hard to connect with just a made up example. Once you're in with your own data that you understand, things make a lot more sense, and I think are a little more intuitive to work with. And we're here for you if you get stuck or need some help. But again, thanks all for joining us today, and hopefully maybe we'll be seeing you again at another session soon. Question here, yeah, not totally specific to the SD, but a question just about the pest codes in general. There's the EPO group. They have like a master database out on the internet that we subscribe to and use. I think we do quarterly updates that we just kind of pull in whatever changes they've made. We kind of pull into our system. But we are a lot more agile than that if you had a specific issue or concern or needed a pest to be added. So each quarterly, we make sure to get any updates of whatever EPO has done out there, but we also have the ability to update our list on the fly if every once in a while we get an email that, oh, hey, this pest is called something different all of a sudden, or I can't find this particular thing in the list, and we're able to kind of work through that. So good question. All right, thanks everybody.